Hello, good afternoon, everyone. Um, Welcome to another week of biology. So we have completed half of the course so far, right? And everything that you just got tested on for test one, you will no longer be responsible for those contents in the second half of the course. Okay, so the final test, test number 2, will just be on the things that we learn from this point forward, okay, the second half of the course, right? And we will have the same thing in the second half. There's going to be quiz 3 and quiz 4. Those will be 10% each. We are going to have a lab. That's going to be also 10%. And then the final test will be 20%. Okay. So starting with the first lesson in the second unit is cellular reproduction. In this lecture, we're going to learn about how the cell divides. And before the cell divides, it needs to uh, copy its genetic material and clone all of its DNA. And we will certainly look at that as well. So let's get started. Lots of things to do here. The general premise is this, right? We learned that in cell theory, all cells came from the division of pre-existing cells. So if we have a parent cell here, parent cell that has, some chromosomes, what we must do before we divide the cells is we must undergo DNA replication. So after DNA replication, we will double the amount of genetic material in the cell. And so when you actually go on and perform the division, the mitosis, right, you will end up with 2 daughter cells which contain the exact same genetic material as the parent cell. Okay, these are chromosomes that I'm drawing, okay? So we call these daughter cells. Okay. So first half of the lesson, we'll focus on DNA replication, and then we're going to do mitosis. So there are a bunch of important scientists that plays a role in discovering either properties related to DNA or structures related to DNA. So let's just go through some of these individuals here. For Chagav, he's the one who came up with the base pairing rule. Okay, so his major finding is that in an organism, the percentage of adenine is the same as percentage of thymine. Right? Similarly, the percentage of cytosine is the same as percentage of G. Okay, we knew this already, right? But we just didn't put a name to it. So Erwin Chargaff, that's the name of the scientist. He basically discovered this, and because of this property, the 2 strands of DNA, are complementary. If you know the nucleotides on one strand, you will know what to expect on the other strand. Okay, so that's Chagaff. Next is Rosalind Franklin. So what she did was she used X-ray to study the structure of DNA. But she died from ovarian cancer before she could finish her work. Okay, but her data suggests DNA is a double helix. Okay. Using her data, Watson and Craig was able to build a scaled model for the DNA. Okay. So using Franklin's data, a scale model of DNA double helix was created. All right, there are two important experiments that we talked about in lesson. First one is Hershey and Chase. Hershey and Chase, they used bacteriophage, which are bacteria that infect, which are viruses that infects bacteria. Used bacteriophage to show that DNA, not protein, is the hereditary material. Hereditary material. Okay. So let's quickly go through Hershey and Chase's experiment in case you forgot about it. So Hershey, Chase. Okay. So they have some bacteriophage, and the bacteriophage are either grown in radioactive sulfur, sulfur 35, or phosphorus 32. Okay. So the radioactive sulfur is gonna label protein because sulfur is found in protein but not DNA, right? And the phosphorus, radioactive phosphorus, it's gonna label DNA. Right? So the bacteriophage has a shell, right? That is made up of protein coating, and then the inside is made up of DNA. So what happens when the bacteriophage infects a bacterium? There's a bacterium right here, and then there is the bacteriophage. Okay. So it's gonna inject the DNA into the bacterium, and then the DNA is gonna get used to make more viruses, basically. Okay. But they didn't know what happened back then. Okay, they knew something was injected, but they wasn't sure if it's a protein that got injected or if the DNA was injected. Okay. So what happens is they would let the bacteriophage infect the bacterium, and then afterwards they would separate them just by blending it, okay, in a blender actually. And then they would separate the content based on weight. So this is a test tube, and there will be two layers. The heavier bacteria along with whatever was injected into them, is going to be at the bottom. And then the viruses, which are much lighter, it's going to be floating at the top. So we have two batches of bacteriophage, those that are labeled with the radioactive sulfur, and therefore the protein shell will be radioactive, but not the DNA. On the other hand, if they were labeled with phosphorus, radioactive phosphorus, then the shell would be regular, but then the DNA is going to be radioactive. Okay, so they let these differently labeled bacteriophage infect the bacteria, and then they separate it and leave it in the test tube. Okay, so for this group, because the proteins are radioactively labeled, that tells you, that the coating is gonna have the radioactivity. Okay, and then they detected the radioactivity at the top, right? Which tells you that the protein stayed outside of the bacteria, right? They didn't go in, right? But that didn't prove that DNA went in. All this shows is that protein didn't go in, right? That's why they have to do this experiment as well. When they label the DNA, and then they separate the viruses and the bacteria, they detected the radioactivity at the bottom. So this shows that DNA went in, and the protein did not. Went into the bacteria, right? They can't just do one and not the other, because in this case, if they only do the label the DNA, radioactive DNA experiment, yes, it shows the DNA went in, but what about the protein? The protein might have went in as well, right? So like, we don't know. But by doing both, they were able to show only DNA went in, but not the proteins. Okay, so that was the Hershey and Chase experiment. Does anybody have any question? All right. Then next we have the Messelson and Stahl experiment. Okay, so these two scientists, they were able to use E. coli. E. coli is a type of bacteria. Okay, so they used E. coli to show that DNA replication follows the semi-conservative model. Semi-conservative model. All right, so what does that mean? Let me just, again, quickly go through it with you. So this one is Messelson and Stahl. So they grew E. coli in medium that contains nitrogen 15. This is heavy nitrogen. So they grow the E. coli in a dish that contains heavy nitrogen for many generations, okay, several generations. So afterwards, all the DNA in the E. coli would have the heavy nitrogen. So if you were to extract the DNA at this point, and then you spin it down on a test tube that separates DNA based on weight, you would expect to find the DNA at the bottom. Right? This is with the N15. So that's the DNA that contains only heavy nitrogen. Okay. Then what happens next is they took the E. coli and then they transfer it to another. These are a petri dish, by the way. That's where you grow the bacteria in. They're going to transfer it to another medium that contains only N14. N14, which is the lighter, right? Lighter, light, or the regular, okay, the lighter nitrogen. And you learn in chemistry, right? Nitrogen has a mass of 14, right? So that they were allowed to grow for 1 generation. Okay, for 1 generation. 1 generation. And then you extract the DNA from it. Okay, so what happens? Well, DNA replication follows the semi-conservative model, right? So what does that even mean? Let's see. If we have the regular original DNA all made up of heavy nitrogen, it would look like this. So in the conservative model, there are 3 possible models at the time. They didn't know which one it was. So the conservative model, okay, conservative model. This is going to look like this. You're going to have the original with the heavy nitrogen, and then when you copy it, the new one is just going to have completely brand new material than the N14. Right? So if that's the case, when you spin it down in the test tube, right, you will have the heavier band, just like the normal, that represents the blue, and then you will have a light band at the top that represents the lighter one. Okay. So that's the conservative model. There is the semi-conservative model. In the semi-conservative model, the DNA separates into its individual strand, and then you will build the other half using new material. Right? So rather than just copying it completely into a new strand, it's half-half. So in this case, when you run it in the gel, where do you expect the DNA band to be? Does anybody want to take a guess?
In the middle.
Sure, go ahead. Where, sorry?
In the middle.
In the middle, exactly. Very good. So it's like a mix of heavy nitrogen 15 and the lighter nitrogen 14, right? So it's going to be somewhere in the middle. And then there is the third model, which is called the dispersive model. And it's very similar to this one, but except for instead of having the new material and the old material restricted in a single strand, they are kind of mixed together. Okay, so each strand has both new and old material. Okay, so like that. And then we have some new material as well. It's kind of interspersed, right? It's dispersed throughout the whole DNA. And if you run this on the gel on the test tube, you will again see one band in the middle because it's mixture, right? So that's what they did. They let the E. coli grow for 1 generation, and afterwards, they did not see 2 bands. They observed this one and this one. So they are unable to decide which one is the correct model. Right? So then they do it again. They do it again for a 2nd generation. So if you do it for a 2nd generation, right? So this is first gen. If you do it again for another generation, for the dispersive model, right, you would expect to see the same thing. Because if this is true, then next time you copy them, it will still be mixed together, right? So you will see one single band in the middle. However, if it is the semi-conservative model, right? What would the second generation be? Okay, so let's take a closer look. Right? This one here, semi-conservative, right? We're gonna separate the two strands, so I will have the blue one, and then I will have the red one. And remember, these E. coli are growing in the light nitrogen medium now. They're growing the red medium, right? So any new material would come from the red one, come from the light nitrogen, right? So in the second generation, you would have a light band at the top, as well as a mixed band in the middle. Okay, so this is second generation. Oops. And this is the observed outcome, and that's how they concluded that DNA replication is semi-conservative. Okay. Well, we can do a quick poll question. Let's see if you are with us. Launch the poll. Okay, go ahead. Okay, not a lot of participation, but that's okay. We will stop the poll, and most people got it right. The answer is B, right? This is to label the protein coating of the bacteria. Here's another one. It's a little bit small. I'm trying to make it bigger here. There we go. Please try this. All right, so let's see. This is saying if the DNA follows conservative model, it's not, right? Like the model is semi-conservative, but the question is hypothetical. It's saying, what if it's conservative model? What would you expect to see? Okay, so you would have the original, and then after one generation, you would have this one, and then the new one, right? Okay, so you see two bands. So immediately I can reject this answer because that's only one band, and I can reject this one. So that and that. And then you got to think about what happens next. Okay, so this one, right? If we let it divide again using the conservative model, you would have the blue one, that's the original, and then you will have the red one, and then the red one would just be red, and then you will make another red one. So you will also see two bands in the second generation. Okay, so the correct answer is A. All right, so good job, the person who got it right. That's the answer. Okay, let's go back to here. Write the complementary strand for DNA below. Be sure to include directionality. So using base pairing rule, we can figure out what's on the other side. If we have A here, then the other side is T, A, G. CC, T, AAT. Just T goes with A, G goes with C. Now, DNA strands are anti-parallel. So if this is 3' to 5' on one end, the other strand is 5' to 3'. Okay. So complementary and anti-parallel. This is a simple question. We've done it several times already. If 23% of all the cell's nucleotides are adenine, what would be the percentage of thymine? Okay, so adenine is same as thymine. So you have 23% adenine, you will have 23% thymine. If you add it up, 23 plus 23 will give you 46. Okay, so A plus T equals to 46%, which means C plus G must be 100 minus that, so 54%. Take 54 divided by 2, then we get 27 and 27. Okay. We're probably familiar with that already. We did it several times before. Okay, cell cycle. 4 stages in cell cycle. So we have the G1 S phase and G2, and then we have the mitosis, the M. Okay, M. So these 3 steps, stages, occurs during interphase. That's when the cell is just, doing its normal thing, performing its normal function, might be growing a little bit, getting ready for division. And then the M is the actual mitosis, the actual act of dividing into 2 cells. So let's take a closer look at this G1 and G2. Okay, G1 and G2. Let me go back a little bit first. The S phase, S stands for synthesis. And that is the step where you actually doing the DNA replication. Okay. So G1 and G2 are checkpoints before and after the DNA replication. So what are you checking for, right? Well, in G1, before you copy the DNA, you want to make sure things are okay. You want to make sure things are in order, right? Before you go ahead and copy the DNA. After all, if the cell is too old, or if the cell is, kind of broken, we don't want to copy the DNA, right? So we're going to check for things like cell size. Cell size, right? Is it big enough to divide? Okay. If it's too small and you divide, then you don't get much cytoplasm, right? And it might not be a viable option. So you need to wait for the cell to get bigger and bigger, and then if it's big enough, then you divide. What about nutrients and resources? Are you actually going to have the nucleotides around to copy the DNA? You don't want to run into a situation where you copy half of it and then you run out of nucleotides, right? Okay. It's like, cooking, right? Before you cook, you gotta make sure you have all the ingredients, right? You can't start cooking and be like, oh no, we don't have potatoes or something, and then you can't finish your dish, right? So G1.
In the video recording, you said the S is the sister chromatin, or I'm the one confused here?
Oh, in this context, S stands for synthesis, and during DNA replication, you will create the sister chromatids. We're gonna get to the sister chromatids in a second, okay?
Okay.
All right. So you gotta check for cell size, you check for nutrients and resources. And then you also will have to check for things like presence, right? And sometimes absence of growth factors or inhibitors. Okay, there are things that promotes a cell division, and there are things that suppresses cell division. So the G1 you gotta check for these things, right? It's not just okay, it's not like an individual choice, right? It's not like the individual cell can divide or not divide on their own. Okay, you gotta take a look at the need of the body, right? And there are these growth factors and inhibitors that would encourage the cell to go ahead and divide, or just wait a little bit, divide later. And then finally, we need to check for DNA quality, right? Are there any damages, right? Any damage? How about telomere length? All right, if you don't know what telomeres are, I'll explain later. Right, so all these things are important. Right? So if all is well, if the cell is big enough, there's enough resources, there's growth factor around, no damage, telomere lengths are fine, then the cell is allowed to progress to the S phase, where you will then do the DNA replication. Okay. So just to address, I think it was Francisca's question. So you have your chromosome. Right? These are the original chromosome. And you have like 46 of them, right? And so if you do DNA replication, then you will end up having exact copy that are stuck together, right? Like that. Okay. So these things that are exact copy of each other, they are called the sister chromatids, right? So all this happens, the DNA replication happens in the S phase. Then afterwards, after you copy the DNA, the cell hasn't divided yet. We haven't undergone mitosis at this point yet. We just copy all the DNA, and before we are allowed to actually divide, we have to have another checkpoint, G2. Okay, it's like getting your driver license. Okay, you gotta take the G1 test and then the G2 test, right? So in G2, again, you have to check for DNA quality. Okay. Any damage again, right? Any mistakes, right? When you do DNA replication, when you copy everything, did you make a mistake? Right? Were you able to correct it? All right. And if there's no damage, no mistake, then the cell is allowed to go to mitosis. So what happens if they don't pass G1 or if they don't pass G2? Right? So if the cell fails G1 or G2, then it will either enter growth arrest. It will not grow anymore. It is still allowed to stay and do its thing. This is called senescence. As we age, more and more of our cell population becomes senescent, and that's part of aging. Or if it's really bad, if the cell has too much DNA damage, and it's just not going to be functional anymore, then it will trigger. You probably know this already. Apoptosis. Okay, like the lysosome might release all of its contents, and then it will kill the cell off because it's just no longer functional. So there you have it, the cell cycle. Any questions before I move on and continue? So if the cell is allowed to go on to mitosis, right, then there is going to be 4 phases, 4 stages within mitosis. So that includes prophase, metaphase, anaphase, and telophase. So the acronym is PMET. Prophase, metaphase, anaphase, telophase. And we're going to explain these later on. Next, we have the all the enzymes that are involved in the DNA replication. I'll just quickly go through it with you. You don't have to draw this down. Okay, I mean, you could if you want to, but it's probably better if you just listen to it, because I already drew this out in the lecture, right? I'm just doing this for those of you who have not watched it, or if you need a refresher. So there is your DNA double helix, a 5' to 3' on one, and then 3' to 5' on the other. And what we need to do is we need to open up a bubble. Okay. This is what we call the origin of replication. All right. We'll focus on this part here. This is the replication fork. Okay, replication fork. And so there will be nucleotides here, right? Like ACTG, TCG, whatever, and then you will have the complementary one on the other side. Right? So I'm not gonna write it out. This is just a quick summary. So 5' to 3', 3' to 5'. Right? So you would have an enzyme here, which is called the helicase. And the helicase is like that, thing that you pull on to zip and unzip your zipper. So as helicase goes down the DNA, it will open up more of the DNA. And so you would have a helicase here, you would have a helicase here. But as the helicase unzip, tension will build up further down the DNA. And so you're going to have another enzyme here that is called a gyrase that will help relieve the tension. Okay, so helicase unzips, but that causes tension to build up, and the gyrase will relieve that tension. If we don't relieve the tension, the DNA might break. Okay? And that's not a good thing. So in order to start the process of DNA replication, you're going to need to have something called a primase. Okay, so this is the RNA primase. And the RNA primase will create a little primer. It's a bit of RNA that's complementary to the parent strand. Okay, so if this is the case, remember the directionality. If this is 3', this is going to be 5', and this is going to be 3'. Okay. The main enzyme that is responsible for building the complementary strand is something called DNA polymerase 3. DNA polymerase 3. Now, this thing has 2 problems, 2 limitations. Number 1, it doesn't know where to begin, okay? And that's what the primase is there for. The primase will put down a few nucleotides. We call it the RNA primer, right? It's like showing it how it's done, right? It's like my kid, right? They come home and say, hey, do your homework. And then they're like, oh, I don't know where to start. I don't know where to start. And I have to sit down with them, show them a couple of questions, and then they will be able to carry on their own. That's what the primase does. The primase will put down a few primers, right? And then the DNA polymerase 3 will be like, okay, I'll finish the job. Right? So that's the first limitation. It doesn't know where to start. So RNA primase solved that problem. The second problem with the DNA polymerase 3 is it can only build DNA in the direction from 5' to 3'. So in this case, it will only be able to build in this direction. That's a new strand. Okay. So as helicase opens up more DNA, it's going to be able to keep on building and building and building. What about on the other strand? Well, on the other strand, same thing. We're gonna have a RNA primer right here. And remember, is complementary. So if the bottom strand is 3' to 5', then what you're building is 5' to 3' like this. Okay? So you will have a primase that puts down the primer, and then the polymerase 3 will come, but it will only be able to build in this direction now, right? From 5' to 3'. That's the limitation, right? So what happens if the helicase unzip more stuff? Well, you're gonna need to have another primer, right? And then you will build some more. And then when you open up more, you will need to have another primer. And then you will build some more, okay? So this top strand here, that is being created continuously with no breaks, that is called the leading strand. Right? No breaks continuously, and it goes towards the replication fork. On the other hand, this one that's being built discontinuously with all these fragments, that's called the lagging strand. Okay? Discontinuously, and then it has all these fragments in it. Goes away from the fork, away from the fork. And these fragments that are created, they are called the Okazaki fragments. Yeah, all this is explained in greater details in the recordings, okay? So if you haven't watched that, you can go and learn it properly. I'm just providing a summary here. Okay, so what happens? Let's take a closer look at some more detail here. What happens when, okay, so we have like a polymerase 3 here again, right? What happens when the polymerase 3 gets to the primer, right? Okay. When polymerase 3 approaches the primer, it will fall off, and then polymerase 1 will take over. Right? I'm not going to draw it. I'm just going to describe it. So polymerase 1 will come and remove the primer, the RNA primer, because the RNA primer contains uracil, right? Okay, and you're going to remove that and replace it with thymine. And all these pink parts, all these RNA primer will be replaced with blue DNA. And finally, we have to glue the Okazaki fragments together, and that will be the job of ligase. Okay, so that's basically what happened. Let's write them down here so you will have it nicely summarized in a table. Okay? Starting with gyrase here. What is going on with the gyrase? Gyrase, remember, is there to relieve tension that builds up in DNA as it unwinds. Okay. If you don't have the gyrase, there will be too much tension and the DNA might break. So the gyrase prevents DNA from breaking. Okay. That is the gyrase. Next, we have the helicase. Helicase. And what does the helicase do? Well, helicase is the enzyme that is responsible for the unwinding of DNA. So unwind DNA, okay, to create replication bubble. Right? And once you create the bubble, the helicase will move into the fork, the replication fork. As DNA is being copied. All right. Now, I actually forgot about the single strand binding protein in my diagram here. Once you open up the DNA strand, the strand sometimes might, have a tendency to come back together because the nucleotides are attracted to each other, right? A wants to bind with T, C wants to bind with G. So we don't want them to close up, right? So we're gonna have these single strand binding proteins. They basically keep the strands apart. Okay, so these are single strand binding proteins. Keeps them apart. Right? So the single strand binding protein prevents double helix from coming back, or we could just say, sure, prevents double helix from coming back together. Keeps them separate, okay? There's a fancy word for coming back together. It's called reannealing. Next, we have primase. What does the primase do? Okay, the primase will create a short RNA primer to help position DNA polymerase 3. Okay. So it helps initiate, I guess should as to these, helps initiate the process of DNA replication. DNA polymerase 3, that's the one that's responsible for actually building the DNA. So it uses the parent strand as a template to build a complementary strand. But remember, there are two limitations here. Two limitations. Okay. One, cannot start by itself. That's why we need the RNA primer, right? We need the primase to start. Right? Do a few nucleotides, and then polymerase 3 can take over. Second problem is that it can only go in 5' to 3' direction. Right? That's why one strand is being synthesized continuously. the leading strand, and then the other strand is being synthesized in fragments, okay? The lagging strand. That's because of the second problem there. Next, we have DNA polymerase 1. So it will replace DNA polymerase 3 as it approaches an RNA primer. And the function of DNA polymerase 1 is to removes RNA primer and adds DNA nucleotide instead. Okay. So an RNA primer may be like this, might be like a UAA. Like a primer would be like UAAGC. And when the DNA polymerase 1 comes, it will remove the U and then put a T in. Okay, remove the A, but then it would just put back another A. So only the U's get to be replaced by T. Everything else should stay the way it is. Now, these two things, the DNA 3 and 1, I should probably make a note here. They have spell checking mechanism, okay, proof reading mechanism. Okay. If they detected a mistake, they will delete it and then fix it. It's just like pressing the backspace on a keyboard when you type a mistake, when you have a typo. Okay. Most of the time, they do a pretty good job correcting it, but sometimes they don't correct it, and we end up having a mutation. Okay, ligase joins Okazaki fragments together. And basically seals gaps on lagging strand. All right. So that's everything that we've done so far.
Can I ask one question?
Of course.
What is Okazaki fragments? Is it?
What are they? Okay, sure. If we go back to our diagram here, right? You see, because the DNA polymerase 3 can only build new strands from the 5' to 3' direction, right? So as you open up more and more DNA, you would need to have another primer, right? To build this part, have another primer. Right? Think about it. If you open up more DNA again, how are you going to copy this part? Right, the top is okay. The polymerase 3 will keep on going into the fork. But how are you going to copy these DNA? Well, you're going to need to have another primer, right? And once you put that primer down, then you're going to be able to, make a copy from there. So this strand is not created in one go. It's not created continuously. Rather, you have to build a little bit at a time. So these little bits of DNA fragments is called Okazaki fragment. It's named after the Japanese scientists who discovered them. Does that answer your question?
Yeah.
Yeah, and that's why we have to join them together, right? At the end, because there's like so many breaks in them. That's what the ligase is for. We join them together. There was another question. Please go ahead. I heard someone else ask something.
No, you answered my question
Oh,
already.
okay, yeah. All right. let's take a quick break. All right, let's take a 5-minute break just for you to catch up with the copying if you need to. And when we return, we'll explain the significance of telomerase and telomeres. Okay, so see you at 1:30. Hello, let's keep going. In order to understand the importance of telomerase, we have to talk about telomeres. Okay, and in order to talk about telomeres, we have to talk about a problem with DNA replication. So let us focus on the lagging strand for a moment here. I'm gonna just draw the lagging strand, right? Not gonna look at the leading strand at the moment. Yeah, so over here, I'm just gonna redraw the lagging strand. And hopefully you understand by now that the lagging strand is going to be created in fragments. So you need to have a primer here, a primer, a primer, a primer, a primer. Okay, so you're gonna have multiple primers, and once you lay down the primers, then DNA polymerase 3 will be able to continue with the replication, right? The replication like that. Okay. And when the DNA polymerase 3 gets to the RNA primer here, it falls off, and then DNA polymerase 1 will come on, and it will remove the primer and replace it with DNA. Okay, and then ligase is going to seal it. Same thing here. DNA polymerase 3 fall off, number 1 gets on, replaces the primer, ligase seals the gap, seals the gap, seals the gap. No problem. Everybody's happy. But what about here? What about at the very end, right? At this location? What's going to happen to this little bit of RNA? Right? We don't have another polymerase coming from above, right? From upstream, we say, because we are at the end of the chromosome. Right? So nothing is going to replace this primer. And eventually, this part of the DNA will just not get copied. Okay, and you will lose a little bit of DNA this way every time. Right? So it turns out, if this is your original DNA, after each replication, it gets shorter and shorter and shorter, and that's not a good thing because you're going to be eating into critical information that are being stored in the DNA, right? Okay, so to prevent this problem, to prevent the loss of important DNA information, at the end of your chromosomes, you will have these telomeres. Okay, these are called telomeres. Right. This is not proportionally drawn. Okay, the telomeres are not as long as the DNA themselves. Okay? And the telomeres, they are tandem repeats. Tandem means one after the other. They're tandem repeats that protects the ends of DNA. Right? So it's just repeats of TTA, GGG, TTA, GGG. It just keeps on repeating itself over and over and over again. And let me see if I have a picture of this thing. Right. These dark blue, those are the chromosomes. And then the little white dots at the end, those are the telomeres. Right? So they're just repeats of, again, TTA, GGG. Just keeps on repeating multiple times. So when we actually go ahead and do the division, right, we will lose a little bit of telomeres every time. So the telomeres get shorter and shorter and shorter with each division, but that's okay because we are not losing valuable DNA, right? So you just, let me just color this in and let's see if it's gonna be better. So there's the telomeres. Okay, telomeres like that. All right, and every time we divide, we're gonna lose a little bit of telomeres. Lose a little bit. Okay, you're gonna divide again, you're gonna lose more. Right? So you're going to keep on losing the telomeres as the cell divides and divide and divide. But you are not losing any critical information. Okay, I don't know why it keeps on coming. It's doing this weird thing. Okay, but you get the idea, right? Like it basically keeps on eating into the the telomeres. Let me try that one more time to see if it's going to go away. There we go. All right. So eventually, if the cell divides sufficiently, then you will run out of telomeres to cut. And if you divide it again, then you would eat into the DNA, right? And that's not a good thing to do. That's why over here, when we had the G1, we want to check for telomere length. Are there still sufficient amount of telomeres left to allow another round of division? If the telomeres are too short or critically short, then we shouldn't divide, because if we divide again, we might lose actual DNA, and that's not a good thing, right? So that's why we have to check it. All right. So when a cell divides multiple times already, it's going to be not allowed to divide any further, and the cell enters senescence. So how do we make sure that the next generation is going to get brand new telomeres, right? We don't want to pass on to our next generation telomeres that are already shortened. Okay, let's say I have kids when my cells have divided like 30 times already. Okay, typically cells divide 40 to 50 times. Okay, so like if my cell divided 30 times already and I pass those cells on to my kids, then they only have like 10 or 20 rounds of division left, right? So we don't want that to happen, and that's what telomerase are for. Telomerase is an enzyme that will relengthens telomeres in germ cells, okay, so that you can pass on brand new telomeres to your next generation. Okay, and there are some cells in the body that rapidly divides, like stem cells, so we would need to have a little bit of telomerase in them as well. Stem cells. Okay, so that's what telomerase is for. It's not found in all cells. It's only found in cells, germ cells. Germ cells are cells that become sperm and eggs, right? And also found in stem cells. Any questions? Fantastic. So that's DNA replication. Now we're going to move on to cell division, mitosis and meiosis. So what have we got over here? Let's see. A is pointing at these X's, okay? So those are chromosomes. Chromosomes are basically condensed version of DNA, and they contains genetical. Humans have 46 chromosomes. Right? B is pointing at these black dots, and that's what's connecting these sister chromatids together. It's called a centromere. Right? This is where sister chromatids are sister chromatids stick together. Next, C here, we have centriole. We first learned about centriole in lecture number 3. They produces spindle fibers. Right? Which is what these lines are for. Okay? The spindle fibers is a kind of microtubules, if you remember. And that's what E is pointing at. Yeah, we're gonna go back. Well, D is just the cell membrane, okay? Cell membrane. But E is the spindle fiber. And the function of spindle fiber is to guide chromosome movements. Okay, it drags the chromosome around, okay, and pulls them to places. So just to reiterate, we have our DNA, right, a double helix. And then what happens is, if we add it with packaging protein, right, some of them are called histones, then you'll be able to take the DNA and then you can wrap it around these proteins. Wrap it around proteins. Okay. So that makes them more condensed. And this is called chromatin. And just before cell division, the chromatins will condense further. Okay. They will condense more. Okay, and then you can copy them. Right, very condensed. And that's the chromosome. Okay. And if they are two same copy, they will be held together at the centromere. And each of these copy is called the sister chromatids. Okay, that's all. Does anybody have any question? All right, so there is a little bit of a misconception here. It's weird that that the like the concept is the way it is. Okay, like it's weird the way that it's supposed to be. And that's why a lot of people get it wrong. I'll explain. Okay. All right. So let's say you start with this. So let's say you have chromosome here. Okay. So how many chromosomes do we see in my drawing here? This is not a trick question. How many chromosomes do you see?
2.
2. Yeah. Excellent. 2. So then we're gonna do DNA replication. Right? Which means we're gonna copy this one and we're gonna copy this one. And after you copy each of them, right, the identical copy is gonna be stuck together, right, at the centromere. Okay, like that. So how many chromosomes do we have now after DNA replication? What do you think?
4.
4?
Is it 4?
Or 8.
4A2. Yeah, I hear several different answers, right? Well, according to the people who come up with this stuff, right, it's still 2. Okay? The rule is this: if they're stuck together, we will still count them as 1. Right? So you started with 2, but even after you duplicate them, this is still considered as 2. Right? So that's 1 right here, and then that's the second one. Right? But we will have 4 chromatids. Okay? So 1 sister chromatid, 2 sister chromatids, 3 and 4. So there will be 4 chromatids. Do you understand?
And when these chromatids are about to replicate again, They would stand as individual chromosomes.
Let me rephrase your question. I think what you're trying to say is, and correct me if I'm wrong, when these sister chromatids separates, right, not duplicate, when they separate and they get pulled
Yeah.
apart, yeah, then we would have 4 chromosomes now. Okay. So
Okay.
if they're not stuck together, we count them as separate things. As long as they're stuck together, though, we count them as one thing. Okay.
Okay, thank you.
Yeah. Everyone else good with this? All right. Now keep this in mind as we talk about the different phases here. Okay, so first we have the end of interphase. What is the end of interphase? End of interphase means we gone through G1, everything's good. We copy our DNA, right? And then we check the quality in G2, no mistakes, no error, everything is ready to go. Then we are at the end of interphase. We are getting ready for the actual division. Okay, so let's write that down. At this point, DNA replication is completed. Okay. And the chromatin, sorry, starts to condense into chromosomes. Okay. What does that mean? Let me just go back here, right? You see chromatins, they are DNA wrapped around proteins, but it's still like really tangled up. Okay, like we can't move this stuff around. You gotta pack it up and move, right? Like physically moving from one apartment to another. You gotta pack up your stuff. Okay, you just can't let them hang loose. So the chromatin is gonna condense and pack up more to become the chromosome. So it's easier to be pulled around. And that's what happens. You cannot see the chromosomes, right? Just like spaghetti noodles, right? Once they become the chromosome, though, it's much more compact that way. Okay, so that's the end of interphase. Remember, by now, synthesis is complete. G2 is also complete. Okay. If it fails G2, you're not going to be allowed to go to mitosis, right? Either going to be like apoptosis or senescence, right? Cell arrest. So none of that. It passed, and it's allowed to divide. So prophase, okay. P for prepare. That's how I remember it. Okay, P for prepare. We are preparing for the actual splitting of the cell. Right? So what do we have to do to prepare? Well, a bunch of things is going to happen in prophase, the first step of mitosis. Okay, so by this point, chromatins completely condense into chromosomes. Right? You can see them clearly now. And then the centrioles, these things, right, will migrate, will move to opposite ends of the cell while producing spindle fibers. Remember, the chromosome is supposed to be inside the nucleus, right? So now it's time for them to get out. And for them to get out, the nuclear envelope starts to dissolve. Okay. So you're just like preparing, you're getting ready for division to take place. Okay. Next is metaphase. M is for middle. Everything lines up in the middle during metaphase. Okay. So the spindle fibers attach to the centromere, sorry, of each chromosome. Chromosomes gets pulled back and forth like a tug of war. And then they line up at the center of the cell. Okay. Center of the cell. So let me ask you, in this picture, how many chromosomes do you see? How many chromosomes do you see in this picture?
4.
4. Okay, anyone else? Thank you, by the way. Yeah, anyone else? How many chromosomes do we see here? You can type it in the chat box if you don't feel comfortable speaking out. Okay. 4 is the correct answer. Right. 1, 2, 3, 4, right? 4. 4 chromosomes. Now, let's trace back. How many did we start with at the beginning? If we have 4 chromosomes in the metaphase, how many chromosomes did the cell begin with? Sorry, how many?
In the video recording, you said chromosomes are the unwind type of chromatin, right?
Okay, yeah, so if we have 4 chromosomes in metaphase, how many did we start with in the prophase?
Same 4.
That's right. It's also 4. It's also 4 chromosomes. Okay. Don't believe me? No problem. Let me convince you. All right. Let me convince you. I know. It's so weird, right? So I'm just gonna use purple, whatever, it doesn't really matter. Okay. So that's 1 chromosome, 2 chromosome, 3 chromosome, 4 chromosomes, right? Okay. So this is like actually like in interphase, right? This is like way before you even duplicated your DNA, right? So you have 4 chromosomes here. Okay. Too long, too short. G1 comes along, cell size good, DNA quality is good, telomere length is good. We will let the cell divide. We'll let the cell copy its DNA. Okay. So after you copy the DNA, now each of them will have an extra one, like that. Does that make sense? All right, so then G2, check them, all good. Then we move on to prophase. So how many chromosomes now in prophase? Well, 1, 2, 3, and 4. Because they're stuck together, right? They're stuck together is still 4. And then in metaphase, we'll have them all lined up in the middle. Right? How many chromosomes do we have? 1, 2, 3, 4. Is that better? For those of you who thought it was 8, have I convinced you that it's not 8, it's 4? All right. Well, I know I convinced at least one person, so that's good. I hope the rest of you understand as well. All right. Moving on. We have the anaphase. What's happening in the anaphase? Well, anaphase, A is for apart. Okay, apart. This is where the chromosome separates, the sister chromatids get separated, okay? So spindle fibers separates the sister chromatids. And pull them towards opposite ends. Okay. Okay, another $1 million question. How many chromosomes do you see in anaphase over here? What's the answer? How many chromosomes?
8.
Yes, this time is 8, right? Because once they separate, they are individual chromosomes, right? So 1, 2, 3, 4, 5, 6, 7, 8. Okay. Now, if you get a question like this on the test, I forgot where my questions are. Is it here? Yeah. Where is this thing? Oh, I forgot where the question is. Practice MC maybe. Oh, there we go. Okay, so here it says, if a cell has 8 chromosomes at metaphase of mitosis, how many chromosomes will it have during anaphase? Let me do a poll. Here, relaunch the poll. Hey, what do you think, huh? What's the answer? Oh boy. Well, thank you for trying anyway. 1% got it right. The answer is 16. 16. Okay. 8 chromosomes at metaphase. So 1, 2, 3, 4, 5, 6, 7, 8. Okay. And then in anaphase, you're gonna separate these things, right? Gonna separate them. So you will have 8 chromosomes, and then another 8. So the correct answer is 16. Okay. Well, you can almost recognize this format, right? This is taken directly from the test. So make sure you understand. And don't just memorize it's 16, okay? Because then what if I ask you about prophase? If I ask you about prophase, then it's 8, right? You really have to work it out yourself and make sure you understand it. Okay, next we have the telophase. T is for tearing apart. This is where you actually split the cell into 2, and you're gonna basically do the reverse of what was happening in prophase. So chromosomes, since we are done moving them, will unpack, will uncondense, decondense, would you say uncondense? Uncondense into chromatids, right? No need to pack them up. And we're gonna reform the nuclear envelope. Okay. Spindle fibers disappear. Okay. And we also have cytokinesis, which is the division of cytoplasm. Okay. Make sure that we have the same amount of cytoplasm for each half of the cell. so that they're the same size. We don't want one to be like super big and then the other is small, right? So that's that. So there you have it. 4 steps involved in mitosis. Now it's a good idea to be able to identify these pictures and know what they represent. So we have some plant cells here. That's the rectangular ones, and then we have some circular ones. So let's take a look at this one. The first one. What stage is that in? Well, when I look at this, I see a single nucleus, and then I can't really see the chromosomes. They're not well-defined X's, if you would. So that tells me this is the interphase. Interphase. This is an animal cells. You can see the centrioles. And the cell is already starting to cleave up and forming 2 separate cells. So that is the telophase. This is a plant cell, and you can see there are no centrioles in plant cells. You might remember that from before, right? But the spindle fibers can still be formed. And over here, we see the spindle fibers. We see the nuclear envelope is starting to dissolve, and we can make out the chromosomes already. So that tells you this is the prophase. Over here, the chromosomes have been separated, and they are being pulled to the opposite end. They're being pulled apart. So apart, A for anaphase. Over here, we have another prophase, this time for animal cell. Nuclear envelope is starting to disappear. You can see the chromosomes. You can see the centrioles moving to the opposite end. Okay, help me out here. What is this next one? What is that? Anybody want to try this one?
I think it's the anaphase.
And that would be correct. Thank you. Anaphase, because they're being pulled apart, right? Everything is in the middle for this one, so that would be the metaphase. And other metaphase here. In your lab, you will have to be able to identify these things, right? And also on the test, of course. This one is going to be the telophase. You can see the 2 separate nuclei that's being formed. And then this is an actual picture. This is onion cell that's being stained. And hopefully you will recognize this is anaphase as well. Okay. All right, let's go through the application question here. This one is just related to the experiment. You can probably do that on your own. Okay, so this here is called karyotype. A karyotype is an orderly arrangement of your chromosomes. Okay, so you can extract the chromosomes from your cells and organize it like this. Which chromosome is the longest? Well, it turns out the karyotype would arrange them by size. The longest one would be chromosome number 1. And then they start to get shorter and shorter until the shortest one is chromosome 22. X and Y are the sex chromosome, and they kind of have their own rules. Okay? Like you would have to be like a cytologist to be able to know which one is which. Like if you haven't, like even if you show me one of these chromosomes, I wouldn't be able to tell you which one it is. Okay, like you have to get training on it, right? So how are the chromosomes numbered? They are actually numbered based on length and position of centromere. Right? Like it's maybe a little bit hard to tell, but there's the centromere. I mean, I can tell that much. There's the centromere, 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 centromere. So depending where the centromere is, that's going to determine the chromosome number. Is this a male or female? How do you know? Male or female?
Female.
This is the
Male.
female. Male?
Yeah, female. XY.
XY, that's right. Y is a male, okay? Females are XX. This one is male. XY. Okay. So it's actually the man that determines the gender of the baby, okay? Because there's a 50% chance that the man will pass on the Y, and 50% chance the man will pass on the X. Right? So if the man pass on the X, then it's going to be a girl. If the man pass on the Y, then it's going to be a boy. Okay. So when I say the man decides, I don't mean like they deliberately decide, right? Like it's like by chance, right? Probability, right? There's a 50% chance of being a male or a female. Okay, but it's not genetically coming from the mother. It's coming from the father. I think I told you, right? Like I have two daughters, and my grandmother, who is very old-fashioned, right? Sometimes she would like pull me aside and say, "Oh, you need to have another child. Something's not right with your wife. She give you two daughters. How come she's not giving you any son?" And then I try to explain to her, it's not her fault. It's actually my fault. We're not having any son, right? Because I am the one who's responsible for passing on the genes that will create a boy. And she's like, no, something's wrong with her. Anyway, I gave up explaining to her. Again, genetically is the male that determines the gender of the child. Explain whether the karyotype show any abnormalities. What do you think, huh? Any abnormalities here? Something that's not normal? Something that's shouldn't be there, maybe? Okay. So here, there's an extra chromosome 21, right? You see? You're only supposed to have 2 of each. So this one is called trisomy. Trisomy 21. And people with an extra chromosome 21 develops Down syndrome. Okay. All right, that's it. Thank you for coming. And if you have any question, feel free to reach out to me through email. Otherwise, I'll see you next Wednesday. Have a good afternoon, guys. Bye for now.
Vincent?
Yeah,
Is
go
he
ahead.
gone?
Mhm.